So in the lymphatic system, we have T lymphocytes and B lymphocytes, named after where they originate. More on that in a bit. But remember that lymphocytes come from lymphoid stem cells, which come from hemocytoblasts in the red bone marrow. And these lymphocytes are responsible for creating antibodies. And the way they function simply is that you have these two working together, and B lymphocytes are the antibody producers. And T lymphocytes, we say that they activate B lymphocytes. We don't get into extensive molecular detail, but let's say that you get a little bacterial bug here. And when this enters your system, your T lymphocytes, they can, in an essence, read out what it is that they're detecting, something foreign. Some of them might directly destroy it right away because they realize, they like their little extensions, their little surface molecules, they're seeing, we're in contact with something that is not from our body. And so they'll attack it. But another thing that they do is that they will activate B lymphocytes, and then B lymphocytes will create a bunch of antibodies to match whatever it is was discovered. And so these antibodies will always seek what the T lymphocyte found in its, while it was patrolling and making sure we weren't getting infected. And so now you have antibodies that will always look for this. And then if this binds to it, we saw what it does to red blood cells, that when antibodies bind to red blood cells, they will undergo hemolysis. This, in one way or another, causes whatever it's attached to be destroyed. And so you can see that now if you have these proteins completely embedding around this, let's redraw it around this little critter here. And once it's there, there will always be some other cells that we've learned about in the previous packets, macrophages that come from monocytes and eosinophils. If we read their descriptions on those tables, you would see that they are white blood cells that destroy antibody-labeled materials. And so that's what part of their job is to do. And this is the premise behind a lot of vaccines, or at least how vaccines used to be done, that when you had a vaccine, you were making a vaccine, you would grab our little critter and then you would shred it apart. Shred him apart. And once he's shred apart like this, what we would do is that we would take out maybe this part, a part that it uses for infecting on the surface, something easy the antibodies can bind to. And then you would isolate this. You would just create 1 million of these and isolate 1 million of these, as many as you can. And then you would introduce this into the human body. And it might sound scary, right? Like, why would we introduce all this dangerous stuff into there? But it's not as dangerous when it's separate from the entire whole. Because if you thought about it, it's like being afraid of a tiger with claws, but no body and no arms. Like, it just doesn't make sense to be afraid of something if it's completely detached. And so it's the same concept, is that these things, we place it in the body, and what that does is that your T lymphocyte will then say, hey, what did I just find in here? And then it'll trigger the B lymphocytes to create antibodies that will always look for this. And why is this so useful? Because now when the real thing comes into the body, you already have antibodies that will attach to that little site that we introduced on the actual critter, the little bacteria or the virus. And then it will be targeted for destruction before it can cause an infection. This is why you can keep getting a cold, because a cold is a virus, a little critter, and doesn't look like this, but it's a little critter that it can change the surface, or like what our bodies are creating antibodies to. So this part will completely look different every year as that cold keeps mutating. And so our antibodies become kind of useless. And so that's the war that we've been having with the microbiological world since the beginning of time. Viruses and bacteria, we keep adapting to each other. We have antibodies that adapt to the new strains, and then these new strains just keep changing, creating new strains over and over again. And so with that, that's sort of our nice little intro into lymphocytes and also what this system is named after. It's named after those lymphocytes. It's named after how we do two things: immunity, how we are able to resist infection and disease. And every single cell, those white blood cells we learned in the blood chapter, they all make up the immune system. And again, this immune system is the ability to resist infection and disease. It's also another definition we added from our chapter 1 topic on those flashcards of organ systems was maintaining blood volume. Blood volume, it's our only liquid connective tissue, and so it contains plasma, and also it has to regulate, I should say that it also regulates the function, the interstitial fluid in tissues. Now, this may feel a little foreign right now, but when we look at our next slide, we'll be able to explain this a little bit better. And so our lymphatic system components are going to be the lymphocytes, both T and B lymphocytes. They are the primary cells of the lymphatic system. Seems kind of obvious now. And again, a little bit of redundancy here. They can destroy, or they are responding to invading pathogens, such as bacteria and viruses. And abnormal body cells, this is in the case of cancer cells, where we talked about certain cancers in this class, and that cancerous cells are cells that cannot regulate their mitosis anymore. They cannot stop dividing. And so even your T and B cells, they can actually create a response to cancer cells, which may have helped us in our day-to-day lives resist cancer, or it can be more harmful in some cases, because our lymphocytes can sometimes become hyperactive and then even begin to think that your regular cells are dangerous. That's what autoimmune disorders are. And so abnormal body cells include cancer cells and then virus-infected cells. These cells can detect whether a cell has a virus or not. And foreign proteins, a little bit beyond the scope of this class, but when you get to microbiology, you'll see that there are some bacteria that secrete proteins in the process of their infection. And so your lymphocytes will detect it and then respond to hopefully deter back that bacterial growth. And then now a new word here, lymph. But this word is something that you've discussed before. It's the same word as interstitial fluid. And remember that interstitial fluid is going to be fluid that tissues share. For example, if we use this next picture on this page, you'll see something that looks very familiar. You'll see the capillaries and you'll see the venules. I think we're all pretty well acquainted with now that blood cells go through here, through the arterial system to arrive to the venous system, and they do so by traveling through these capillary beds. But what also flows through it is plasma that contains nutrients, gases, and so these little cells here are little fibroblasts. Maybe not this one, but fibroblasts. And they are responsible for producing collagen fibers, elastic fibers, and even reticular fibers. And so they need nutrients. They need oxygen from the respiratory system. And so gases escape this area, nutrients escape this area, but also remember that these cells in the process of using those nutrients, they will return waste. They're going to poop right back into those blood vessels. And they're going to produce a waste into this fluid. This fluid that they're all swimming in is shared. This word is what interstitial fluid is. And so when you see the word interstitial fluid, you can see that it's not much more different from the word plasma. And plasma contains electrolytes, nutrients, and even waste that other tissues have placed back in the blood vessels, and gases. And you go out here, what's the difference? There's still gases. These are producing CO2. Oxygen made its way into this fluid, interstitial fluid. The nutrients are in here. There's wastes in here. Gases, I think I just said gases, electrolytes in here. And so there's no real difference too much, or there's no big difference between interstitial fluid and plasma. And so now today we add the word lymph. And the word lymph exists only in these lymphatic vessels. This is a third set of vessels that, as common as it is to see a photo of people with blood vessels, the red and the blue ones, the arteries and the veins that pump blood in and out of the heart, you have this lymphatics, these lymphatic vessels. You have a third set of vessels that maybe is not too popular in the mainstream world, that you kind of have to take an anatomy class to find out about. And they regulate liquid volume. And how they do this is that if we look at this, is that between all exchange sites, between wherever there's capillaries, and remember there are capillaries in absolutely every organ system. In the integumentary system, they are in the dermis. In the muscles, every muscle has, you see how many, you know how many muscles we have from your lab exam that you just took. All of them have capillaries. And so through the capillaries, exchange areas, there will be lymphatic vessels that take in fluid in one direction. And this lymphatic fluid will then exit, and we'll talk about where it goes after. But when the fluid goes from interstitial fluid out here and then goes in here, it becomes the word lymph. But at the same time, it's kind of the same material at the end of the day, right? It's nutrients, wastes, gases, things like that flow right into it. And so with that, we go back to where we leave off. Make sure we know when we start calling it lymph. The difference between interstitial fluid and lymph. Now with this, we're going to see that the lymphatic system contains lymphatic vessels, which we just simply call lymphatics for short, instead of having to say lymphatic vessels. And remember, they contain lymph. And they have two sites of entry. They have right there at the tissues, and then finally, the veins. And this will come up later, like 2 slides from now. And then this slide, this has to do with certain organs and structures, like for example, that play a role in lymphatics but don't necessarily occur in the lymphatic vessels. Some of those that are a little more common, like tonsils, thymus, spleen, and then we're going to see in digestive, we're going to cut this off here for this chapter, but when we get to digestive, we bring this back. But even in your digestive tract, respiratory tract, urinary, reproductive, there are these groups of lymphoid tissues that have lymphocytes right there guarding the epithelial tissue. And so we'll talk about that a little bit later. But then also the appendix, and then even red bone marrow is considered part of the lymphatic system because it is the site where lymphocytes originate. It's where lymphoid stem cells come from. And then all of these ducts, don't worry, I know it looks very intimidating, but we're going to cut out a lot of this for our exam. And so we'll talk about what makes something primary and what makes something secondary as we keep going. But what we're going to keep in mind as we move forward is that the primary lymphoid tissues are where lymphocytes are formed or mature. But this is not the same as being on, like an on switch. They're made, but they're not switched on yet. And so that's the main difference between primary and secondary. Primary lymphoids are formed and mature, but in secondary lymphoid, you'll see that's where they are activated. So it's kind of like they're waiting to be activated, exposed to something dangerous. The primary lymphoid tissues, they're going to include obviously where they come from, red bone marrow. Remember in our blood chapter, which is coming up in next week's exam, that there is myeloid stem cells and lymphoid stem cells. Today's topic cares about lymphoid stem cells. And then lastly, we have our thymus, which we'll talk about a little bit further. And so we move on to this slide, and then we add a little bit more detail about these vessels. And so remember that when we are discussing words like tissues, remember, yes, these blood vessels are technically made of tissues. They have endothelia, they have smooth muscle, probably some other connective tissues there. But now the highlight here is that I want to make sure that you understand when we say the word peripheral tissue, that you are looking at only the space that I'm highlighting here. It is the space of the fluid where the interstitial fluid is. That's what peripheral tissues are. All of these cells are tissues when you combine them together. The word tissues means many cells working together. And so that all those cells can represent anything. Right now it's fibroblasts because I think it's labeled as fibroblasts. See, it says areolar connective tissue, so I just assumed fibroblasts, but that's mainly what it's going to be. And so, but this is every organ that you see that needs blood supply, that has capillaries attached to it, can be considered a peripheral tissue. This word means outer tissues, outside of the blood vessels. And we're going to see that the lymphatic system, and it's kind of, we're talking about it right now. So let's talk about this point next. And so we say venous system, only veins. So they carry lymph from peripheral tissues to venous system. Now we're going to have to jump just off of this point to our next slide here before we add that detail, all the way to this slide. And then we'll finish this one before we move on. And while you're here, what I need you to register is that you are seeing two blood vessels here, the veins, which I know that in lab you haven't been taught these yet, but this is going to be called the brachiocephalic vein, almost going into the subclavian vein. But the main take home of this is that you'll notice that these lymphatic vessels attach to the veins. That's what we mean by venous system. That no matter where it came from, right? You have capillaries here, right here with the muscles, the internal and external intercostals. External and internal intercostals. They're going to have capillaries. And then that lymph fluid is going to flow through these vessels, travel up, travel more, and then be placed right back in the veins. And we know that these veins are going to do what with blood? In what direction are they taking blood? Towards the heart. And what major vessel is this one going to be then? Yes, the superior vena cava. So this is going to lead into the heart. And then the heart will continue to pump blood through the arteries, pumping whatever went through this lymphatic vessel. And so these two drainage sites of the veins, where they drain into the veins, one is called the right lymphatic duct on the right side. And then we can see another one that, should have been conveniently called the left one, but it is called thoracic duct instead. But we'll see why in a second, why there's some sense to it. And so considering the cutoff, because the lymphatics is something I kind of want to just lightly touch on for our exam, because I know it's a big exam. So you don't need to know all of these vessels, but I do need you to know two facts about the thoracic and lymphatic duct. That if we look to the right here, you will see another image that sort of is a mystery as far as how humans are built, is that the thoracic duct is called thoracic because it mainly serves all of the thoracic area, the abdominal area. It just seems to be the major duct because of all the fluid flow in that lymph, all that lymph flowing through the vessels, the 75% of that fluid returns to only the left thoracic duct. And the last 25%, and again, these numbers are estimates just to give you a gauge where the majority of flow goes through, that lymph returns mainly on the left side where the heart is. You do not need to know this, but if I asked you the majority of lymph fluid drains to which duct, that's the major take-home. So here in parentheses is our take-home. Majority of fluid drained into this duct. And remember, fluid should be appropriately called lymph here. So you can see why what's in lymph, what's in interstitial fluid, what's in plasma really just differs on concentration of oxygen, maybe some waste materials going in one, nutrient levels, because they're all picking up and leading it somewhere. And if we're leading into the veins, you can see that when would you be able to tell the difference between lymph and plasma in here? And so and we'll see why they have to flow through here instead of just going through the venules like we saw in the previous photo. But before we go back, we make sure to remember majority of the body's lymph returns to the thoracic. The right lymphatic duct gets less of the body's lymph. I don't know another way of expressing it. The other one, the one that gets less. And I add this again as estimates just to make sure that you understand why. And I love this picture down here because I just kind of broke a human being into quarters there. And so remember, maybe make a note to yourself, you only need to know the major ducts. You don't need to know all of this. The lab will add some of those on the final lab exam. All right. And so with that, we go back to where we were. And now that we got the big picture of where it's going, so you can see that all tissues will guide and lead lymph will flow through here and it will be guided to either the thoracic duct or the right lymphatic duct. And these vessels are made up of something that should look very familiar to blood vessels. And they are found very close to the capillaries. And therefore, they are tiny little vessels before they get very large. And just like with these blood vessels, we call the tiny blood vessels capillaries. These are called capillaries as well. We call them lymphatic capillaries. And remember, that means small vessels, the smallest vessels possible, capillaries. And if you look at a major difference between the two photos, when blood plasma flows through here, it's not like in lymphatics where they flow in here in one direction. So these vessels have a connection, a continuous connection. They are connected to something. These are not. These are dead ends at the tissues. And then they all will guide it to those two ducts, the right thoracic and the right duct and the thoracic duct will guide that lymph and it'll flow in one direction to those veins that we mentioned earlier, or to those ducts. The lymphatic capillaries are therefore closed at one end. And the good news is that if you studied blood over the weekend a little bit, I know you had your lab exam, but if you did, you'd remember that what was the name of our simple squamous cells found in blood vessels? Endothelium. And good news, guess what the ones inside of capillaries are called? Endothelium. I'm looking for it labeled, but I'm not seeing it. Right here, sorry. Overlapping endothelial cells. And so we can add that highlight there. And one thing that they do is that they create this unique structure that prevents backflow. In this class, what are objects that prevent backflow? Valves. And so valves, this valve here is just made from the cells overlapping. You can see overlap, 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 overlap. And then these are going to prevent backflow of lymph. So once the lymph flows in, or I guess once the interstitial fluid flows in and becomes lymph, then that lymph will flow, go through these valves, and then make its way through those ducts that we mentioned, the right duct and the thoracic duct. And guess what's waiting for any possible invaders? Lymphocytes. Now, lymphocytes are sitting in here, and this is why you have this system. As you never know that in your digestive tract, maybe you brought in a little virus from something you ate. And so that little virus makes its way through that endothelial cells, and now maybe he's starting to infect your fibroblasts and fibrocytes. Not the same thing. And then so as we're looking at this cell is now infected. And maybe there's more little viruses being released, but some of those virus particles will go in here. And lymphocytes, once they detect something foreign, they produce, now what do they produce? antibodies. And so they produce antibodies, not really drawn here, I'll draw it for you, but this is one form of the antibodies, like that. And they'll send them out, and then they'll now look for little virus that came and infected you. And now they can spread that antibody quickly through your lymphatic vessels, and then for better protection. And so we had some of the notes that we mentioned have overlapping endothelial cells. Their overlapping creates one-way valves. You don't need to know what things are flowing in and out of the lymph. Just assume it's the same concept, same thing as plasma. Nutrients, waste, water, and even maybe other nutrients. But I feel like that might make it overwhelming. But just keep in mind, it's circulating whatever's in interstitial fluid. And then it's going to return to plasma. So there's not just minor changes in what you've learned, that what's in plasma. Either high nutrient, low nutrient, high oxygen, low oxygen, high CO2. So I'm trying to make it less overwhelming there. And then when we say the word dead cell debris, this word means dead cell, like dead cell parts. Like maybe there's a mitochondria completely that left a cell that died, that formed, that underwent autolysis, self-destruction. And then those little parts are floating around, and now they'll get rid of it. The white blood cells get rid of it. So that allows them to flow into areas with white blood cells. And don't forget, one-way flow. All right. And now this is sort of where we sort of segue really forward. So we kind of went to the end of the story there, but all that lymph is going to flow to the body's trunk. And remember, it's going to enter the right duct. Let me reposition my iPad here. Right duct plus thoracic duct. And remember how veins relied on our skeletons for some sort of contraction in order to passage lymph through it? Then here, contraction of surrounding skeletal muscle aids flow of lymph. So in your heads forever, always remember artery and vein, and don't forget, there's always a lymphatic vessel with them as well. Here's a cool little microscopic image of a valve as flowing through here. And then once that fluid makes its way through, this closes, and then it doesn't allow any fluid, but maybe back here, but it gets stuck here. Matches this one here. All right. And so with that, we move on. This page, let's keep it going. And let's keep it right here very quickly. Yeah, you're not going to need this page. Unless you guys want it. Now the last thing to add about these duct systems is that we remember our right lymphatic duct and our thoracic duct. There's one last little structure that matters, and this is sort of a major site of where lymph drains from the lower body. And so this is called cisterna chyli, like the actress or the singer. I'm not sure what she is, the model. The Pepsi one, cisterna chyli. And then so Kylie, and it's spelled like this, like Kylie a little bit, but it's Kylie. And that's all you need from this page. And so this says drains lymph from inferior body before draining into thoracic duct. All right. And so as we look at this, and I'll go ahead and highlight with you on this one. All right. All right. And so now we go into our populations of cells, and we have our lymphocytes. And remember that lymphocytes come from lymphoid stem cells, as I terribly wrote up there. Lymphoid stem cells. And there are two directions or two development pathways or two differentiation pathways, meaning that they can turn into different things depending on where they are located or, yes, where they're located. And so here on this side, you can see that if we are in the red bone marrow, that's where we derive B lymphocytes, which we call for short B cells. These B cells can then produce antibodies, and then they can morph, or I'm going to say differentiate. I always like to talk sci-fi with these for some reason. But otherwise, when you look at these, they're going to differentiate into plasma cells. And these plasma cells will release tons of antibodies. That's their main job, is to produce tons of antibodies at any moment that they get activated or they have to because of an infection. You then have natural killer cells. That's what NK stands for. And they do exactly what they sound like. They are going to look and patrol for things that don't belong. They're going to engulf it and destroy it. And so those are called our natural killer cells. But you can see that red bone marrow was their site of development. If you look at this other side, the thymus, this is why we call all of these T lymphocytes, T cells. Because they instead develop in the thymus. In the thymus.
And the senses, are they going to call it lymphocytes? B lymphocytes? Or?
I might use either or. Yeah, good with both. And then when we look at cytotoxic, helper, regulatory, memory, there are different T cells that do different things, either together or directly they attack something, or they don't directly attack it. Like, for example, cytotoxic T cells have directly attack. These will activate these and say, hey, I need you to go attack some stuff. Or these will go, hey, I need you to produce antibodies quick. I see something there. That's what their job is kind of to do. And then there's other ones that are there patrolling, making sure things are okay. And then like this one, for example, this one, when you've been infected, it stays around. And then it'll always be looking for that thing that got you sick that last time. And then if it enters your body one more time, then it says, oh, it's back. And then it'll go and trigger the B cells. Then these will probably turn to plasma cells. And then you have a huge antibody storm to attack whatever infected you before. And so that's why we call these memory T cells, because they remember older infections by keeping the antibodies necessary to destroy whatever it was that infected you previously in your life. But they do have a shelf life. And this is why some vaccines do require an update, because without that update, your memory T cells probably might have already said, well, I haven't been infected by this for about 5 or 6 years, so I guess I don't need to be here anymore. And they kind of disappear. And that's why you need to refresh vaccines. Some of them last longer than others. The reason behind that, I don't know, might get you a Nobel Prize if you figure it out. Now, with that, T cells, B cells, and natural killer cells, these populations of cells, overall, though, they can kind of be summed up in how they defend us. And T cells are the most abundant, so we write most here, and then least down here. To remember, because we should know which ones are the most abundant lymphocytes, which ones are the least abundant. And T cells are going to be responsible for cell-mediated immunity. And that means that they are going to control the intensity of the immune response. And let me rewrite this, actually. Control intensity B cells produce antibodies, and those antibodies therefore give it the activity of antibody-mediated immunity. So you can see that it's kind of telling you what exactly is giving you that immunity. And an example, remember that immunity is your ability to resist infections. So antibodies do that for you here. The cells themselves do it here. And then natural killer cells, these are going to be immune surveillance, right? Like security. So I have here patrolling security. But these security, they'll engulf something dangerous.
Why do you talk about memory and
Because antibodies serve as memory. Like those antibodies will stay in your system. These T cells will trigger these B cells to produce that antibody that you need to fight off that infection. And so this is why we kind of can say B cells contribute to our long-term memory of immunity. And don't forget that the word antibodies and the word antigens, like we saw in blood typing, they go together. Antibodies, at the end of the day, they're proteins. In our blood chapter, we call them immunoglobulins. Proteins looking for antigens. That's their job. And antigens are anything that pretty much cells looks like something that cells bind to that don't belong. But I will say that the word can be used in a different... I think that the perfect way to describe it is that anything being sought, that means like looked for, is the best word for antigen. Because think about autoimmune disorders. Your own white blood cells can attack your own cells. That means that your own cells serve as antigens. And you can see why I don't call it foreign, because what happens in autoimmune, they're attacking you, and then the antigens are what you're already made out of. And so we say antibodies that look for antigens. So antigens are anything that can bind to antibodies is kind of the best way. Say again?
Antigens can bind to what?
Antigens always are bound by antibodies. Antibodies bind to antigens. And then so here, this word, I like that it's here most and not all antigens, but most antigens are pathogens, proteins, foreign proteins, abnormal substances. And so antigens, in the context of immunity, it means anything that's going to stimulate an immune response. So you can see an autoimmune, if your white blood cells are attacking your own body, your blood cells, your own body becomes antigens in some spots, some areas. All right. And then some quick functions for our 4 little T cells. Our cytotoxic T cells, the name cyto, cell, toxic T cells, they attack foreign cells such as bacteria or body of cells infected by viruses. And this type of immunity requires direct contact. And then there I have control the severity of the immune response to simplify a lot of what we're going to say. And so helper T cells, as I keep describing with our little story, that these are ones that are there to say, hey, I just found something, right? Because these can go directly to something dangerous and say, hey, time to get rid of it. But then these, what they do is that they're the ones that I mentioned in our little background story about if you're introduced into something bad, that these, and now I guess I can add this here, helper, that these cells will then go and say, hey, I found something. You should probably create some antibodies for it. And then the B lymphocytes will say, all right, let's create a bunch of antibodies to look for that. And so helper T lymphocytes are that very, or do that very activity of activating B cells, but they can also activate other T cells. The only thing to add there. And so again, my main favorite one though for our segment is for our intro to anatomy, activate B cells, and they can produce antibodies, right? Because they've been activated. That's why I use that story in the intro. Regulatory T cells, they just moderate the immune response. And again, that means that they might tone it down, increase it, and that's way beyond the scope of this class. So it moderate or control, regulate immune response. Memory T cells, these are going to be, again, these cells are ones that were created. Like let's say these helper T cells, they detected an infection. You created the antibodies and you survived it. Then some of those T cells will then will turn into memory T cells, which will then, they'll stimulate stem cells to turn into ones that will remember just the one that infected you. And then these are memory T cells. They'll always be able to detect the bacteria that infected you in the past. So they respond to antigens they have already encountered. And so with that, we move to a little bit of where their development occurs, and that kind of leads into organs. Now, it is my hope that you've seen the chapter or answered the study guide question on myeloid and lymphoid stem cells. And so remember that in red bone marrow, you will have cells called hemocytoblasts. And hemocytoblasts originate and differentiate in bone marrow. Oh, in bone marrow, sorry. And they can differentiate into myeloid stem cells or lymphoid stem cells. And so our topic in lymphatics doesn't care about myeloid stem cells because myeloid stem cells become either red blood cells and all the other white blood cells we learned about in this class. The never let monkeys eat bananas, those. And then except L, right? L is lymphocytes. And so lymphoid stem cells will eventually turn into lymphocytes.
So we don't need myeloid?
You need it for the blood exam. For the blood part of your exam. And so right now our focus is not on the myeloid stem cells. And so as we look at this pathway for lymphoid stem cells, if they stay in the bone marrow, they develop into B cells and NK cells. So that's what this note is trying to tell us. If stay in bone marrow, they turn into B cells and natural killer cells. And then I like this little table because it tells you exactly what even their immunity categories in, where B cells produce antibodies, and immune surveillance is done by natural killer cells. And if we go to the thymus, which I know that some of us might not have seen our thymus before, but we'll see it next. That's going to be an organ, a lymphatic organ. And that's where, if lymphocytes, the same lymphoid stem cells that became these, if they migrate to the thymus, they become T lymphocytes. And so that's the take home there. And then the T lymphocytes are responsible for cell-mediated immunity. And so B cells produce antibodies. When they get stimulated, they become plasma cells and they secrete antibodies. And I just put in an insane amount. Because B cells can produce antibodies, but then when they turn into plasma cells, their main goal in life is to produce that antibody in question, the one that you need. And just a repeat of what B cells do. And natural killer cells, exactly what the name implies. Attack foreign cells, virus-infected bodies, and cancer cells. This says antibodies for cancerous cells, bacteria, and viruses. So you can have antibodies for all of those things. For cancer cells, bacteria, or viruses.
What does that say on the top? Something amount?
Insane amount.
Oh, insane amount.
Yes. And this entire process, right, as you can see my bracket here, is called lymphopoiesis. Those lymphoid stem cells we learned about in the blood chapter, they produce all the lymphocytes within the two groups. It just depends on where they migrate, and that's what points 1 and 2 are about, what we just mentioned. One group migrates to the thymus, T cells, red bone marrow, and they produce B cells and natural killer cells. And so to finish up this chapter, we really just have nodules and organs to talk about, but we're going to go very light on it, I promise, because some of these are going to come back, so I'm going to part it halfway. And so, first thing we got to do is know the difference between a nodule and organs. And nodules and organs are essentially the same exact thing. They are huge, they are sacs that contain lymphocytes inside of them. The only difference between the two is that an organ has a capsule around it like this one here. That's the only difference, is that when we look at lymphoid nodules, there's going to be no fibrous capsule surrounding that container, that sac, that cluster. No fibrous capsule. Lymphoid organs such as the spleen and the thymus, these are going to be examples of lymphoid organs. And they are going to be surrounded by a capsule. Now, this is something we're going to bring back in digestive tract, but something to keep track is that this is an example, is that this is a cluster of white blood cells, a cluster of lymphocytes, I should say, lymphocytes. And they are there in the digestive tract waiting in case that you're eating, you're passing your nutrients are going through the epithelial tissue, the columnar cells with microvilli, that's going to be these little tiny ones right here. And then underneath the epithelial tissue, there's a basement membrane. And then underneath that basement membrane, there is going to be the connective tissue layer. And in that connective tissue layer, you can find capillaries, lymphatic vessels, but also you find these so that if a little virus or a little bacteria that you ate, then the lymphocytes are ready and to attack whatever might have been absorbed into this space. And so this is an example of a nodule, a lymphoid nodule, because there's no capsule around it. It's just a cluster of cells, and there's no capsule. But it's an example. We'll bring this one back in digestive tract, but it's a good one to introduce. Next. This one is also in the digestive tract and essentially almost is exactly the same thing. A little cluster of lymphocytes on the defense. But again, you don't need it until we get to digestive tract, then we add it. But the one that we do need is the tonsils, because there's no other time in class to talk about it. And so the tonsils are going to be the large lymphoid nodules, not organs. They don't have a capsule. And they are these little, and I'm going to keep it nodules. Nodules are lymphocytes patrolling for invaders. And so they are there. If we look at our illustration, this is the site of where most bacteria can enter your body, whether through while you're eating, because of breathing. And so these tonsils are an example of clusters of lymphocytes. So that's what your tonsils are. When they get infected and swollen, is because they were exposed to something that an antigen, either from a bacteria or a virus or maybe even a cancerous cell in that space, but more than likely bacteria. And here, the one that's located on the top of the pharynx, the superior portion of the pharynx, the nasopharynx, is the pharyngeal tonsil. Pharyngeal tonsil. If we remember our hard palate is made by the maxilla, and then the last 1/3 approximately is made by the palatine bone. And then we have our soft palate. And so palate, right? That's where this one comes from. Palatine tonsil. And so palatine tonsil. And lastly, lingual tonsil is going to be, lingual in Latin means tongue. And so lingual is going to be at the base of the root of the tongue. Lingual tonsil. So we have those 3 tonsils to worry about. And they are an example of lymphoid nodules because they do not have capsules. And tonsillitis is when you get inflammation of either one of those tonsils. But don't worry about this word. I know that you've all heard it. I know you know what it is.
What does it say there before that on the lymphoid nodule location?
This one right here?
No.
This?
No, more to the down there.
Deep to mucosa, which I don't expect you guys to kind of know what mucosa is quite just yet. In the past, I put this chapter after digestive tract, and so these notes are reflecting that.
You don't want us to know any of that?
For this chapter exam, no, but you will need it later. It's in another packet in your digestive.
But that what you put in the square, we don't need it, right?
Yes, you don't need what's up here either,
It's right at the bottom either.
but you do need this one.
But where do we put that?
This right here?
Yeah.
Yeah, you don't need it. All right, and we are almost done. Last one to add. I know this has a lot here, but this is another little bit of a cutoff. The nodes. This is a lab concept where you're going to have to know the names of the nodes, and that's good enough for this introductory anatomy. So as we look at nodes, when you look at your blood vessels, and I got to show you another picture. Stay on that slide, and then but here, look at all the vessels, and then look at every single there's some areas that are going to have nodes, and these are going to be little clusters or little bundles of clusters of lymphocytes. And so think about how cool this is, that these capillaries, the lymphatic capillaries, are bringing all that lymph back, draining it into the subclavian and on both sides here, the veins that are here. We haven't gone over vessels yet, I forget. But veins here. And then this vein that drains on this side, another one that drains on this side. But before they get there, they have to go through all of these clusters. So that lymph is traveling and it goes through a cluster. Traveling, goes through a cluster. And so it's bringing back fluid, and it has to go through all those clusters. And those clusters, at the end of the day, what they are is that they are small lymphoid organs. And if you're an organ, that means that you have a capsule. So I hope by now you know the difference between lymphoid organs and nodules. And be careful, because the word nodes and nodules sound eerily similar. So nodes is actually a lymphoid organ, nodules are not. And these are the example, these are the tonsils. And nodes are not. So it sounds like it, but it's not. Nodes and nodules, not the same word. It's something that I encounter gives some issues. And so remember, nodules do not have a capsule, and nodes have a capsule because they are an organ, have capsule. And their job is to filter and remove all the pathogens, essentially all, from lymph before that fluid can return to the right lymphatic duct and the thoracic duct.
Can you go up again?
All right. I might bring this node back when we get to our urinary chapter, but right now don't worry about it. This is all we need. And so with that, we move on to the next page, to our next lymphatic organ. And then we have our thymus, that famous thymus, which has a role in lymphocyte development where T cells develop. And I guess I could write that shorter here. Let me.
You don't want us to know that, or we'll go back to it?
Sorry?
You don't want us
No, you got to know that. You got to know nodes. Role in development of T lymphocytes, T cells. Located in the mediastinum. This will help you in lab. And the good news is that that's all you need. We're not going to do histology. 35, Anatomy 35 will go through that. But otherwise, lymphatic organs of thymus play a role in T lymphocyte development. And so with that, we then move on to the spleen. And our spleen is going to have a few things that sound familiar. First, it's going to have a hilum. Remember that the hilum is going to be a structure, an indentation where objects like blood vessels, nerves, and lymphatic vessels can enter. The respiratory system, the root of the lung, the hilum, is an indentation where the bronchus and the major blood vessels, pulmonary arteries and pulmonary veins, enter and exit the lungs. Here, hilum, it's a indentation where lymphatic vessels, veins, and arteries can be supplied to the organ.
What did it say? Red pulp?
Yes, and then so here, when you look at the inside of this organ and you look at, say, this image, you'll see the word red pulp and white pulp. On a microscope, you would see that these white pulp contain lymphocytes, these red pulp contain red blood cells. And so red blood cells, red pulp, white blood cells, white pulp. And then the last thing we need here is that what is the role of those pulps? Is that remember that this is like a filtering mechanism. If all you have is white blood cells, it is doing, well, actually, it's doing two things, but doing a few things, but two things that we should know is that it has white pulp, therefore, that means that blood is flowing in, and also lymphatic vessels, and all of that is being patrolled, or also a huge organ for a lot of space to patrol and remove any pathogens that are going through it. And also something slightly to touch on here, we might remember this, that when we look at this portion here, I'm going to make sure that we find it somewhere. When we look at this part, when we saw red blood cells, we have to recall that the average lifespan of a red blood cell is 4 months. And so it's circulating, delivering oxygen for you, and then eventually look what organ it makes its way into. The spleen. And so this can happen in the liver and bone marrow, but spleen. And so what you have in the spleen is that you have macrophages that recycle red blood cells. You can see that they break apart red blood cells, and then that heme group, that iron-containing group that carries oxygen, is then taken back, or the parts of the heme group are recycled, iron, and then they will be transported all the way to become red blood cells in the red bone marrow. And so the spleen becomes a site for recycling red blood cell components. And so not only is it involved in lymphatics, right, immune responses, we're going to have to write here that macrophages in spleen recycle red blood cells. White pulp contains lymphocytes, and we know what lymphocytes are there to do. They're supposed to be little clusters ready to elicit an immune response in the case of an infection. All right. And that's it for all this. I know it has a lot of text, but you don't need a lot of it, just those major take-homes.